Genetics reading. In 1843, at the age of 21, Gregor Mendel entered a monastery in Brünn, Austria, where he was given the task of tending the garden. In 1851, he was sent to the University of Vienna to study science and mathematics. When Mendel returned to the monastery, he used his knowledge of statistics to analyze how traits, physical characteristics like height or flower color, are passed on from parent to offspring. The phenomenon Mendel was observing is called inheritance, the passing of traits from parents to their offspring. Mendel's experiments. To see whether there was any predictable pattern to inheritance, Mendel decided to experiment with the inheritance patterns of pea plants. Mendel studied 7 traits of pea plants. The traits he studied were flower color, seed shape, pod color, pod shape, plant height, and flower position on the stem. Additionally, each trait had contrasting forms. Flower color could be either purple or white. The plant could either be tall or short. The patterns he found form the basis of our knowledge of genetics. He began by growing plants that were peer breeding for each trait. Plants that are peer breeding for a trait always produce offspring with that trait. Pea plants that breed peer for the trait of yellow pods always produce offspring with yellow pods. Mendel obtained 14 peer breeding pea plants, 2 for each of the 7 traits. He called each strain a parental generation, or P1 generation for short. Mendel then transferred pollen from a plant pure for one trait to the flower of another plant pure for the contrasting trait. This exchange of genetic information is called cross-pollination. To do this, he dusted the pollen from a yellow-podded plant onto the flower of a green-podded plant. Then he allowed the seeds to grow. Next, he recorded how many of each type of offspring each P1 plant produced. Mendel called the offspring of the P1 generation the first filial generation, or F1 generation. Then he self-pollinated the flowers from the F1 generation. Mendel dusted the flowers of a pea plant with pollen from the same plant. Self-pollination involves no exchange of genetic information. Then he collected and grew the seeds. Mendel called the plants on this generation the second filial generation, or F2 generation. Following this process, Mendel performed thousands of crosses and documented the results of each. Mendel's results and conclusions. In one of his experiments, Mendel crossed 2 plants, one pure for green pods and one pure for yellow pods. The resulting seeds produced an F1 generation with only green podded plants. No yellow pods developed, even though one parent had been pure for yellow pods. Only one of the 2 traits found in the P1 generation appeared in the F1 generation. If Mendel had concluded his research at this point, he might have interpreted the results to mean that crosses between green-podded plants and yellow-podded plants always produced offspring with green pods. However, Mendel self-pollinated the F1 plants and then planted seeds from the resulting F2 generation. When they grew, he observed that about 3/4 of the F2 plants had green pods and 1/4 had yellow pods. The apparently lost trait had reappeared. After studying the results of these crosses, Mendel developed a simple set of rules that govern inheritance of traits from parent to offspring. His rules are summarized below. The rules of inheritance. Number 1. Mendel suggested that each parent plant offered packets of information to the offspring. The packets of information were in the gametes. Mendel created each packet of information a factor. Mendel's factors are now known as genes. Number 2. Each individual has 2 genes for each trait, 1 from each parent. However, the genetic information in those genes may or may not be the same. If the information inherited from each parent is the same, the individual is said to be homozygous for that trait. If the information inherited from each parent is different, the individual is said to be heterozygous for that trait. Number 3. Different forms of a trait are called alleles. The alleles an organism possesses are called a genotype. The appearance of an organism is called a phenotype. Number 4. The offspring receives 1 allele for each trait from each parent. The copies are passed to the offspring by the gametes. He decided that during the formation of gametes, the 2 factors in the parents separate. Mendel concluded that each gamete receives only 1 copy of each factor. Number 5. The presence of an allele does not ensure that the trait will be expressed. If an organism is heterozygous for a trait, only 1 allele will be expressed. The one that is expressed is called the factor dominant, since it masked or dominated the other allele. The dominated allele is called recessive, since its presence was masked. Chromosomes and genes. Most of Mendel's findings agree with what biologists now know of the structure and function of genes and chromosomes. However, when Mendel presented his findings in a paper he wrote in 1865, scientists had not yet discovered chromosomes, nor did they possess any knowledge of meiosis. However, in 1900, biologist Walter Sutton proposed the chromosome theory, which linked observations of meiosis with Mendel's principles. Biologists now know a gene is the segment of DNA on a chromosome that controls a particular hereditary trait. Because chromosomes occur in pairs, genes occur in pairs. Each contrasting form of a gene is called an allele. Scientists use letters to represent alleles. Capital letters refer to dominant alleles and lowercase letters to refer to recessive alleles. For example, the allele for the dominant trait of tallness in pea plants is represented by capital T, and the allele for the recessive trait of shortness by lowercase t. The allele for the dominant trait of a green pod color is represented by capital G. The alleles for yellow pod color by lowercase g. References for this reading are from Curtis and Barnes' Invitation to Biology, published by Worth Publishers Incorporated in 1981. Also Johnson and Raven, Biology, Principles and Exploration, published by Holt, Reinhardt, and Winston in 1996. Finally, Tolley, Modern Biology, published by Holt, Reinhardt, and Winston in 1989.